morning, everyone. Thanks for being here. I don't think Dr. Lass needs an introduction, so Dr. Lass, you can go ahead and get started.
Thanks, Sam. Well, I've given grand rounds for many years to this department, and I would say this is the most personal lecture I've ever given. It really will give you some idea what has made me tick over the years to drive my interest in clinical trials. And I taught a lot of classical music to our residents over the years in surgery, but I haven't had that opportunity for many years. So you'll get a little touch of my musical side from this lecture as well. I've been thinking about putting this lecture together for many years, but I had the opportunity in September as part of the World Doctors Orchestra, which I'm in. We gave a session in Copenhagen, and they organized a music and medicine seminar, and to the staff at the main hospital in Copenhagen. And I was one of the lecturers for this. So this gave me an opportunity to put this together, and I thought it would be great to present this also to our department. I have no disclosures relevant to this presentation. I just wish to acknowledge the support we've had from the National Eye Institute for all this work, the Cleveland Eye Bank Foundation Oversight, and I openly express my love for Brahms also. The common theme, and what I'm hoping to give you is some sense of the common themes of Brahms, who was a famous romantic German composer of the 19th century, and his art, and my science over the years that have in many ways has been inspired by his art. And I think these are, whether it's science or art, I think there's creative elements to both. And you're not content with the status quo, but while being inspired by the past, you want to drive to answer new questions. No question, you have to have perseverance and a determination to succeed, and discipline in learning the trade. Role models are really important, and you want to collaborate because you can't do it yourself, and you really do need to learn from criticism. And have a short-term strategy where you know you can succeed. Have some short-term risky things you might be doing, but also strive towards those longer-term big things. And Brahms, we'll first talk about Brahms. He was born in Hamburg and had many influences from teachers, in Hamburg. And probably the most notable influence he had early on was by a very famous violinist of the 19th century, Joseph Joachim. And then ultimately Joachim introduced to one of the most famous composers at the time in Germany, and also a critic who really promoted music in Europe, which was Robert Schumann and his wife Clara. And but he was heavily influenced certainly by his predecessors, including Bach, Mozart, and particularly Beethoven, who he had obviously huge respect. And the highest mountain to ascend and go beyond was the Ninth Symphony that Beethoven nearly ended his career with. And once he was introduced to Schumann, Schumann was very impressed by Brahms. He was in his 20s at the time, and wrote early on in one of the journals that Brahms was fated to give expression to the times in the highest and most ideal manner. I mean, he really set the bar that Brahms was going to be the next great German composer. And he really pushed Brahms to write a symphony early on, even in the 1853. But Brahms was not ready. He was not ready. And he kept putting Schumann off. And he actually wrote that I shall never write a symphony. You have no idea how it feels to hear behind you the tramp of a giant like Beethoven. But as you may know, you can express different feelings, emotions in the key and this is certainly established by Mozart and Beethoven. When you want to have more of a sad, reflective thing, you would have a minor key or more dramatic whereas a more joyous or pleasant key or celebratory key would be in a major key. Now, Beethoven's Ninth Symphony started in the key of D minor. And so he had that key, Brahms had that key in mind, and Schumann was pushing him to write a symphony in D minor to follow up with Beethoven. And he ultimately did experiment with that in a number of different pieces, but he ultimately did not write his first major work for orchestra as a symphony, but as a piano concerto, his first concerto. But he was very much influenced by his mentors from the classics. But he wanted to establish his own identity, but through smaller works, either on piano alone, solo, or chamber music. And he ultimately, obviously, a major influence was moving from Hamburg to Vienna, which was the heart of music in Europe at the time. So he went through a progressive process of different orchestral works to move towards the symphony. And he wrote the serenades, the concerto I mentioned, a work for orchestra and chorus, the German Requiem. He wrote Haydn variations, which are very popular. And finally, the initial sketches for the first symphony were in 1854, pretty early on. But he really held off, as I said, until he started thinking about the symphony more seriously in 1868, when he was somewhere in the Alps and heard this horn. And he quoted, "Thus blew the shepherd today." And I'll just play what that horn sounded ultimately in his symphony, which introduced the transition from the minor key, that serious key, to the final big positive major key in C major in the fourth movement. And hopefully this is going to play. So that definitely announces that something really positive is going to be coming, and I'll play where it finally went to from that. But that horn inspired him to really move towards completing the symphony. But he was fighting, as in science, there's tremendous competition, and he was competing with a different group of composers that were going in a completely different, they were not tied to the past. They were very much what would call the radical progressives, coming up with a different concept, not based on the normal classical structures of Haydn, Mozart, and Beethoven, but going in a completely different direction, including the French composer Hector Berlioz, the Hungarian composer Franz Liszt, and the German composer Wagner, who concentrated particularly on opera. So there was a tremendous competition between the classical conservatives like Brahms or Schumann and these other groups. Ultimately, we both benefited from that competition. The music is very different, but at that time there was an intense competition in the music world over this. But he completed his symphony in 1876. Now he was still very concerned how this symphony would be received. Instead of having this premiere of this symphony in like the major city, Vienna or Berlin, he decided to choose a small town in Germany to premiere his symphony, because he was worried about its reception. And he knew the conductor very well. in Karlsruhe, and that's where the first symphony was premiered. He was kind of testing it out. It's like a Broadway musical. He kind of wanted to test it out in Cleveland before he took it to Broadway. So that's what he did with the symphony, and ultimately it was a resounding success. And as I said, the composition was very much based on the Beethoven model, where the first movement is a very serious, in a minor key, the conflict of life. And then it takes you to the great ending, similar to Beethoven's Nine. Here's the beginning of the first movement. Now, this is, let me go back here. This is what you call absolute music. He did not have any specific story in mind, as the Romantics did, like Liszt and so on, where what's called a symphonic poem. He wanted each person to take their own personal experiences and react to that music. So when you heard that serious opening of that movement with that percussion, bum, that beat there, each person may react to that, the seriousness of that first movement. At the same time, people would react similarly in the positive sense with the ending of the fourth movement after that horn solo, which was the big theme which he was criticized for, taking Beethoven's Ninth Symphony, da 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 da, and this is what he turned it into. You know, when I did it in a room, it was a live audience. Anyway, it was very similar to Beethoven's Ninth, the last movement, and he just basically inverted that theme to reverse it to create that theme. His symphony was, as I said, well received in Karlsruhe. He then took it on to the Munich and Vienna, etc., and then it went around the world. It was hailed as Beethoven's 10th Symphony. Bill Roth, the famous surgeon, general surgeon, was a very good friend of Brahms, and he wrote that. In fact, Brahms dedicated a number of his works to complicated chromatic themes, inexorable timpani, as you heard at the beginning. Last movement seems to be one of the greatest and most glorious of your endings. It is one, and then Hanslick, another critic, said, is one of the most distinctive and magnificent works of the symphonic literature. Now, this is at the same time that this symphony premiered Wagner, his arch rival, premiered the Ring Cycle, which is one of the great cycles of opera in the literature. Now, the other point I want to make is it took him 20 years to write that first symphony. That second symphony only came a year later, only a year later. And the point is, once you know how to do it, it's a lot easier to do it again. And so he wrote a very positive second symphony, very much reflecting when he composes at an Austrian retreat. And then he went on to write 2 more symphonies, 3 concertos, and 2 operas. So this was a process that he went to over 20 years to get to being confident to write these symphonies and be confident that it would be well received. Now, as far that's my musical side. I'm going to go a little more quickly here because we do want to give Loretta time as well for her talk. But I'm going to give this more. You've seen a lot of these slides before a couple of years ago when I've given my clinical trial talks about the results of our NIH trials, but I want to do this more in the context of the inspiration of art in terms of clinical trials. I also had a wonderful group of mentors at BU and at Mass Eye and Ear. But I was particularly not only influenced by Brahms, but also by Monet, who basically had his entire creative life outside his window in his garden. And he basically perfected over when he moved to Giverny in the late 1800s and created Giverny. He really was able to perfect and advance his art form of Impressionism throughout that. And so he basically had a niche, a style, a technique that he continued to advance in all his years until some of it deteriorated with his cataract problem. But I started as a medical student. I had, as I said, a great role model with Dr. Leibowitz, and in doing animal models, primarily looking at inflammation in the cornea. And I went to my first ARVO meeting in 1972, and we published in 74. I didn't do any research as a resident, was really busy clinically. I did a lot during my fellowship with all the mentors that I had there. But I joined here in 1979, and I really didn't know what I wanted to do. I obviously had a lot of experience in animal models, but I'd also been involved in clinical research. So I kind of started doing both corporate research, where I was a investigator in an industry-sponsored study, not my own ideas, but just learning the trade of working within a clinical trial with a defined protocol, and also was doing animal models for onchocerciasis that ultimately got funded by the Eye Institute. But again, the same thing with mentors, getting a sense of the field, focusing on a subspecialty, attending meetings, listening to who were the most successful in the field, what was their interest, and developing the different methods as a clinician scientist, including how you write an abstract, a paper, poster, how do you give a talk of different lengths, courses, etc. All these things are different methods for developing your eventually your own style and how you communicate with your colleagues. So as I said, I was involved with corporate trials. Certainly a great way to get started is with a case report, and then your own ideas with a single site prospective study, and then you need to ultimately find that niche. Well, like Monet was Giverny. For me, it was the corneal endothelium. And particularly with the influence of Dr. Dolman, who was one of the first to describe the endothelium and its function as both its pump and barrier function. And this was a great time because we were doing a lot of transplants with I'm glad Bill Reinhardt could join today with my colleague Bill, and also there was tremendous changes going on with corneal storage, where it had gone from whole globe to MK medium, and then ultimately to intermediate storage out as long as 2 weeks. So there was a lot to study on the clinical trial side for transplants and the storage of corneas. And I even developed this a factors slide as a fellow of all the different aspects, and I've added to that over the years as procedures have changed, of all the factors that could influence transplant and cell loss changes. And the ones that ultimately I was able to be part of or lead was areas where you could actually do a randomization. And obviously it's hard to randomize on the recipient side, but it was fairly straightforward to randomize on the donor side for factors that really was influencing practice, whether it was the method of storage, tissue matching, the age of the donor, how long you preserve the corneas, and now most recently diabetes. But then you could study all these factors if you planned it accordingly in a prospective way. So my first real area of design of a clinical trial was with the corneal storage solutions, where there were all these solutions coming out. We knew the standard. How would the new solution compare to the standard? So we were fortunate working with Bill and myself, and ultimately we reached out to other investigators like at Mayo Clinic, that you could do a paired design where one cornea would go in one solution, the other cornea would go in the other solution, and you would do the same procedure back to back on patients with the same diagnosis. So it was a very beautiful paired design. It allowed us to do a relatively small sample size. And we did a whole series of these papers in the late 80s, early 90s that ultimately led to Optisol GS, which is still the predominant storage solution on the market, my goodness, 30 years later. Their Light Force, unfortunately, had to be withdrawn because of manufacturing issues. And this also, to couple with the clinical trial side of thing and its coordination, we formed the Reading Center for tracking endothelial cell loss following these procedures. I was very inspired by Walter Stark and Maureen McGuire that did their tissue matching, which was a very hot topic at the time for these high-risk transplants with matching, with HLA matching. This ultimately did not show that matching made a difference, but it certainly showed that eye banks and surgeons could work together to answer an important question. And what I learned from talking to the Eye Institute is that you need to have a common theme within you need to initiate, these were the different steps in terms of moving towards a multi-center trial. And I was fortunate to get involved after they had initially conceived of the study of the cornea donor study, where they were looking at the effect of donor age on transplant success. There was a tremendous prejudice of using corneas over 60 years of age. And Mark Madison at Holland came up with this trial. We ultimately joined them, and then Beth Ann joined us as well in the early 2000s, looking at age effect on graft outcomes, both success and cell loss. It was a large study, and they randomized on the basis of 12 to 65 years versus 66 to 75 years of age. It was a large study, as I said, 105 surgeons, 80 sites, 43 eye banks. And this was around 2000. At 5 years, there was no difference in graft success, 86%. But we continued the study for another 5 to 7 years because of the cell loss data. Interestingly, there was a difference on at the younger and oldest age subgroups. And so ultimately, the way this was presented to the community was that there was similar graft success from 34 up to 71 years of age. And that's ultimately what was accepted in the eye banking and surgeon community. The reason why we went beyond 5 years was that there was a significant difference in cell loss at 5 years, where both had substantial cell loss, but it was less for the younger group under 65 than the older. But of the surviving grafts by 10 years, there was no difference. So what I learned from that trial in talking to the Eye Institute was that you need to have a simple question that can be answered, that will have major impact 5 to 7 years from the time you conceive of that question and to answer. You need to have a measurable outcome in a defined period of time with a reasonable sample size. So usually recruitment has to be completed within 1 to 2 years to allow sufficient time, because most funding occurs within 5 years and not any longer. It needs to be an answer that will be useful to impact practice patterns. And you can't study, normally you can't, they don't want to fund a procedure, studying a procedure. They want to, which might change. And certainly in transplants, that has been the case. You need to answer a more fundamental question around eye banking and transplant in this case. And in terms of how you would define a clinical trial, there are 4 elements. You have a treatment group and a control group. You have the randomization, you have the masking of both the patient and the researcher, and the ethics have to be in place for, and obviously to have the appropriate equipoise, that the investigators are comfortable that the question has not been answered and that they're willing to enter into that trial with the patient to answer that question. So the next trial we did was looking at the effect of how long the corneas were preserved after death to surgery. And we knew there was a tremendous prejudice using corneas beyond 7 to 8 days, whereas the exported tissue out of the country, we were using out to the full FDA-approved time of 14 days. So, and obviously answering this question would give more time, and extending beyond 7 to 8 days would give more time for the eye banks to evaluate the tissue, distribute it, and obviously expand the donor pool with threats that could occur from infection, etc. So this was, as I said, my first symphony that I composed, and it was conceived of in 2007. And ultimately, it was funded in after failing the first time in its review. It was funded in 2011. So this was 23 years after my first consideration of running an RCT in the late 80s. So it was similar to Brahms's experience, and Brahms was always on my mind as I was going through writing the grant, getting it reviewed, failing, and then ultimately resubmitting and having it funded. And I see the Cornea Preservation Time as my first symphony, and I've told some of my collaborators like Loretta and Beth Ann that I saw myself as the composer and the conductor. The orchestra, I had one of the finest orchestras in the United States, with 40 clinical sites, over 60 surgeons, 23 eye banks. We were funded by the National Eye Institute principally, as well as different organizations, while Brahms was funded by his publisher, Simrock. And my publisher was all these journals, and obviously Brahms got his out through Simrock. And we were fortunate to work with the group at the Jabe Health Center, and ultimately these were similar elements that we'd had in the cornea donor study. This was a similarly large study with 1,330 eyes, randomized for donors between 7 days to 8 to 14 days. And ultimately, we came up with all these papers showing that preservation time showed similar success out to 11 days. So that extended about 3 to 14 days. And the cell loss data also confirmed that. And then we were able to do all these secondary analysis, looking at other factors for transplant success. One of the signals that came out of that was that donor diabetes, the corneas from donors that had diabetes did not do as well. They had more cell loss, they had more failures, and they had more complications early on. So as a result of that, like Brahms, I immediately knew, now that I knew how to write that first symphony, we then proceeded to start working on the second symphony, which is our current study, the Dex trial. Now, I had more trouble than Brahms getting it funded. Then he was able to get his symphony out in 1 year. It took me from the idea of doing this trial, it took us from almost 9 years from when we first conceived of this to finally getting funded in 2021. I knew how to write it, I just couldn't get it funded. So, but anyway, we have a great team now with Dex, and obviously diabetes has a major impact, huge around the world, and 40% of our donor pool is diabetic now. So we need to know which donors we can use for the transplant, which ones we can't. And so similar to the CPTS, we're looking at graft outcomes with a randomization. 2/3 of the corneas are coming from normals and 1/3 from diabetic. And we're also checking the donors to make sure they are normal by doing postmortem A1C testing to make sure that their A1Cs are below 6.5. We have 28 active sites, over 50 surgeons, and 13 eye banks. And we're, again, tracking all the different factors besides diabetes. This is where we stand as of yesterday. We did our first surgery in February of last year, and we just hit our 1,200th eye enrolled last Friday. We're approaching 1,000 surgeries performed. So we just have a great group of clinical sites that are accomplishing these surgeries. This shows our timeline for enrollment. We've hit our timeline for enrollment now, even though we were affected by a number of factors during the with work issues at different sites, etc. But we're well on track for completing our surgery enrollment by this coming spring, and we're working with Dr. Monnier in the Department of Pathology to look at AGE, advanced glycation end products, in the donors to see how that correlates with graft outcomes, which is sort of a novel thing to look at besides diabetes, yes or no, or the A1C. And we hope to finally get our results out by and that premiere of the symphony truly when it gets published in early of 2026. This will allow us to give a more targeted approach to the use of these donor corneas. I want to just finish just with a few minutes that one of the dreams that came true in 2009 with my love of Brahms and also the love of my colleagues, both doctors and musicians in the World Doctors Orchestra, was bringing the orchestra to Severance Hall in 2009, and this was one of the great moments for me and for Cleveland to bring this international orchestra to Cleveland and benefit the Cleveland Free Clinic playing Brahms' First Symphony. And this is Dr. Willig, who is our conductor, based in Berlin, who is a cardiologist and an excellent conductor. And I just want to play the ending of the symphony for you. All right. I'm sorry I might have gone a little over, but I hope you enjoyed my my scientific and musical connections here, and I will turn things over to Loretta, who's been my great colleague over the years, supporting a lot of my a lot of all this work. Thank you.